So we've done the math for calculating transport across the membrane. Now what we want to do is get into a little bit more of the kind of conceptual movement and how different molecules move across the membrane, because it's not always going to be an anion or cation moving across the membrane. So we need to get into, well, what does it look like? So transport of molecules may either be mediated or non-mediated. So non-mediated would be something like movement simply across the membrane without any sort of membrane protein. Okay, non-mediated transport, simple diffusion. So it can simply move across the membrane without any sort of outside influence. Now, the rate of diffusion depends on the concentration gradient and the substance's solubility in the membrane's nonpolar core. Okay, so, well, how soluble is that molecule? So this is generally going to be reserved for and most commonly seen with simple molecules and arguably kind of your essential molecules like water, O2, and not really shockingly, but steroids can diffuse across the membrane quite readily. Okay. Mediated transport falls into two different categories depending on the thermodynamics of the system. Okay, so you have passive mediated transport or facilitated diffusion. So molecules are going to flow from high concentrations to low concentrations. And active transport is one in which a molecule is transported against its concentration gradient. This endergonic process must be coupled with an exergonic process to make it thermodynamically favorable. So we've got non-mediated transport and then mediated transport that falls into the category of our facilitated diffusion, or active transport where we're moving something across the membrane and we're using ATP in order to basically move it across that membrane. As an example, we use ATP. Now, for both simple and facilitated diffusion, solutes only move in thermodynamically favorable step, or thermodynamically favorable direction. Now, this graph right here on our y-axis is our rate, indicated by v for velocity, and our x-axis is going to be our substrate, okay, and our substrate concentration. Now, when looking at this graph, and looking at specifically this curve right here, our facilitated diffusion curve, is there a graph that we've seen previously that resembles this? Well, we've actually seen two. We've seen our oxygen binding curve, and we've seen our Michaelis-Menten plot. Okay, so proteins, which again, if you think about facilitated diffusion, well, our oxygen binding curve was a relationship between a protein's ability to bind oxygen and at what point was it saturated. And then if you think about this similarly to a Michaelis-Menten plot, well, what concentration of our substrate basically saturated our enzyme and it couldn't go any faster? Well, here we've got proteins can facilitate the transport and increase the rates of transport. Okay. Now, this is quite important, or there are some important distinguishing features. Facilitated diffusion requires assistance generally from a protein. Okay, so we'll get into some examples where there's facilitated diffusion that is not reliant or dependent on the protein, but generally when you talk about facilitated diffusion, you are talking about something that is protein dependent. Now, this transport displays saturation kinetics, which harkens back to our discussion of a Michaelis-Menten kinetics and Michaelis-Menten plots. So once saturated, the only way to increase transport is to increase the number of transport proteins, because remember, what that x-axis shows is our concentration of our substrate, and as that substrate concentration increases, at some point our curve just plateaus. It goes, and then it plateaus. However, if you add more proteins, well then it goes up. Now, mediated transport here, so we've got largely protein-mediated transport. Proteins are going to facilitate that transport, or facilitate the transport of molecules across the membrane. These proteins are going to also increase the rate of transport. Now, the solutes can either go along the concentration gradient through facilitated diffusion, or they can go against that concentration gradient. So whenever we talk about something going against its concentration gradient, what we're talking about is, well, first of all, we're talking about active transport. We're going from low concentration to high. If it's going along the concentration gradient, it's going high to low. And these are the conventions that you have to kind of adjust to and be comfortable with. All right. Now, there are a number of different types of proteins that are involved in this transport. There are channels and there are carrier transporters. So protein channel, like if you think back to whenever we were talking about this stuff in when we were talking about membrane proteins, well, this would be like one of those beta sheet proteins that basically opens a cavity and enables these small molecules to transport across. Whereas a carrier protein has basically three different phases: A, B, and C. In phase A, it's going to be open to the extracellular space, enabling the small molecule to bind. In B, it's going to undergo a conformational change, where that small molecule is no longer accessing the extracellular space, and it's not accessing the intracellular space. And then in C, now that protein's undergone another conformational change, where that small molecule is moved into the intracellular space, and it's free to kind of exist within there. So channel transporters have a pore through which the solute passes at a very high flux rate. They are very selective, and these rapid flux rates tend to be gated. And we're going to talk about different gate proteins later, but voltage gated or small molecule gated or something like that. Examples are porins, gap junctions, and ion channels. We're going to get into some of these examples a little bit later. Now, carrier transporters bind solutes on one side and undergo a conformational or structural change, releasing the solute on the other side. These are divided into pumps and transporters, also known as translocases and permeases. Now, carrier transporters show temperature dependence, while channel-forming transporters do not. So, that's worth recognizing, that carrier transporters show temperature dependence. Okay. So what this figure here on the right-hand side of this slide shows us is the synthesis of serotonin. So we've got tryptophan going to serotonin. Serotonin is packaged into these vesicles. These vesicles merge with the cell membrane, enabling the serotonin to be released, and ultimately that released serotonin binds with a receptor protein on a receiving neuron. The space between these two neurons is known as a synapse, but also what's unique about these is that these receptor proteins, well, what they're going to do, these serotonin receptors, is they will bind to serotonin, but ultimately they will release it. That released serotonin goes back to the neuron that released it originally. And there are these proteins known as serotonin reuptake transporters. And what they're going to do is, as you can follow these little red dots, is they're going to basically take that serotonin back up. That serotonin is going to be packaged into vesicles. Okay, so this is an example. This serotonin reuptake transporter. This is a transport protein. So what we have is these transporter proteins make absolutely great drug targets because the serotonin, whenever it gets packaged, so the serotonin can be taken up and packaged by the vesicle. Well, if you block this, what happens to that serotonin? Well, that serotonin ends up out here, in which case it can bind to these receptors, in which case you're stimulating, you're basically saturating those receptors. Okay? Serotonin reuptake inhibitors are transporter proteins that are targeted for drug, or that are generally going to be drug targets. These transport inhibitors can, not this serotonin in particular, but they can cause disease or death. So examples of those would be carboxytractolyside and boncrenic acid. These are inhibitors of mitochondrial ADP ATP translocation. So the movement of ADP in favor of ATP. These are two examples of inhibitors that basically disrupt that reuptake. Now, the thing is that these two different drugs as inhibitors of these two, these are great drug targets, not necessarily from the, you obviously do not want to synthesize something that is going to do this to human beings or anything like that, but if you have this ADP-ATP translocation protein, you could target this as, something that's fatal to like a parasite or to some sort of, to a bacterium or something like that. Alright, well I hope this is helpful, and I'll see you in the next video.